Okay, so let's get started. Hello everyone. Welcome to our first time 2025 Immune Zoom seminar. So first of all, I wish everyone online have a happy new year. And it's really my pleasure to welcome our New Year's first speaker, Dr. Lishan from University of Maryland, opened the 2025 Immune Zoom seminar series. So, so Lishan currently is a chair, endowed professor for HIV research and a full professor in pharmacology, microbiology, and immunology in University of Maryland School of Medicine. So Lishan, I just to give a very brief introduction about Lishan. Lishan got his PhD degree in biology from Harvard University and then followed with his postdoc training in immunology at Stanford. After that, he worked as a senior scientist in a biotech company which called Systemic Synthesis, which is current now Novartis, mainly working on HIV pathogenesis and blood stem cell based HIV gene therapy in humanized mice and patients. Um, but after that, Li San actually moved back to academia and then joined as a faculty member, joined as a member in Department of Microbiology and Immunology, School of Medicine, University of North Carolina, Chapel Hill, all the way up to full professorship. From 2021, Nissan moved to University of Maryland as the Charles Gordon Smith Endowed Professor for HIV Research. Also, he was appointed as the director in Division of Viral Pathogenesis and Cancer Institute of Human Virology. Meanwhile, he also was appointed as a full professor in Department of Pharmacology and Microbiology and Immunology in University of Maryland School of Medicine. So Li San's lab is mainly interested in studying human immunity and inflammatory disease, and then to develop antibody and cell-based drugs targeting novel immune cells and the signaling pathway. His lab study how HIV and HBV interact with human immune cells to cause inflammatory diseases. And then his group has discovered and focused on the pDC interferon axis, which I think we're going to hear today, in the immunopathogenesis and therapy of chronic HIV infection and other human inflammatory disease. His group has also started investigating the pDC interferon macrophage axis in tumor microenvironment and in cancer immunotherapy. Additionally, his lab also developed novel candidate drugs, including antibody, CAR T, and therapeutic vaccines to treat human inflammatory disease, which including virus infection and cancer. So really appreciate Lisan's joining us today to share his science with us. And then we're very much looking for your talk. Please go ahead.
Thank you, Tran. Good morning and Happy New Year to everyone. It's a great pleasure to open the year and to talk about what we have been doing in the lab for the last decade or over 10, almost 20 years, working on how HIV causes disease. And in the last few years, we actually transitioned after I moved to Maryland from HIV therapy to other human disease with no HIV infection, including cancer. So today I'll give a report what we have been doing, and I think I met Tran a few months last year in CI. That's how we caught up, and I heard about this seminar series and have been hearing and joining this series. I'm really impressed by the speakers and have been enjoying the speak. Uh, Today, so I'm going to really follow what I have diagram here on the bottom right, study how HIV interact with a cell called plasmacytoidendritic cell, or really should be called interferon-producing cell, IPC. Then they convert the cell into this red cell, like now called disease-associated pDC or inflammatory pDC. And this lead to many disease caused by HIV, including immune suppression and tissue injury. So we have been working on this pathway to really dissect the pathway using HIV as a tool. And in recent years, we showed that in tumor microenvironment, some tumor, the same pathway seem to be induced in the tumor. And therefore, some of the similar approach we are trying to treat HIV disease, we're trying to develop into a tumor immune therapy. So I'll give you update what this about, and hopefully I'll be able to finish most of them, if not all of them, I have outlined here. First, I'll give you a general introduction about how we reached the stage where we think the disease-associated pDC and interferon, type 1 interferon, is a major inflammatory pathway in multiple disease in, in human health. So I also just give you some recently or unpublished data about our study of treating HIV-associated end organ disease. As you know, HIV patient on the ART usually do not die of AIDS, but they develop some other disease in other organ. We call it end organ disease. That's really from head to toe. It's like premature aging disease. So the second part, I really want to focus on the immune mechanism. How does this pathway induce immune suppression for HIV in the lymphoid tissue and for tumor in the tumor microenvironment? So the two pathways we recently discovered, one is the metabolic dysregulation, mitochondrial activity impairment by pDC. And the next part is about really how this pathway may target the really important CD8 T cell called stem-like T cell. That's the cell recently really reported to be important for controlling tumor and controlling chronic viral infection. The last part, I just give you a transition where we have been doing in the lab to study how this pathway really can be used to study other inflammatory disease, including MASH, so metabolic dysregulation associated steatosis and hepatitis, including fibrosis and liver cancer. Last part, if I have time, I'll show you an effort to really study how this disease-associated pDC program tumor microenvironment and cause all the immune suppression in tumor microenvironment, many of them, and how we can target this pathway for immune therapy. So to just remind you what's a good immune response should be based on really decades of work from studying mice, mostly with acute virus infection. So that's from Charlie Janvey's immunobiology textbook. You can see the green line is after virus infection. You get the mostly type 1 interferon and the probably activation of the IPC. Then you have the blue one line, the innate immune cell get activated to provide antiviral activity and to program the T and B cell in the red line. And then the yellow region is the viral load or the antigen load will disappear as the red T cell, B cell come up, they disappear. Then you have a T cell memory. One thing to notice is really the green line and the red line do not overlap much. And that's the kinetics and magnitude of type 1 interferon have been shown to be really critical in coordinating the whole immune response in a building. However, during the persistent presence of antigen, the yellow area does not go away. That's including chronic viral infection, cancer, and the autoantigen like autoimmune disease. What happened to this green line, blue line, and red line? How do they call this pathogenic response is what less clear. We have been using the HIV persistent as model to study this condition to see how this misregulated green line will affect the red line and the other pathogens. So HIV is really provide a good model to study human inflammatory disease, as it can show here with ART in HIV patient. They can live more or less the normal age of uninfected patient. However, they develop many non-AIDS-defining end organ disease from head to toe. We have a neurologic disease, you have a metabolic disease, heart disease, cancer, lung cancer, lung, liver, and hematopoiesis. So it's really a model where we can study how this inflammation induced by HIV contributed to this different disease. And today I'm going to really give you some aspect how we study this aspect and using the liver disease model as a way to show how this can be targeted for immune therapy. The goal is really to clarify inflammation. So in this case, we call inflammation HIV. In this case, what's the effector cell? How does the effector molecule work? And what's the mechanism? Can we target them? So to study HIV and the human immunity, really, the challenge is HIV only infects human cells. So in this case, we have been using the humanized mouse model pioneered by Michael McKean in Irving Weissman's lab in the late 80s, where he transplant human fetal liver stem cell and the human thymus and get this liver model. And over the years, you have second and third generation humanized mice. I listed here as a review we put together a few years ago. You can see the commonly used is really the hematopoietic stem cell only in the ASG or NRG mice. This is the easy one and easy to make. The most sophisticated one is really the thy-live version in the better mice with a bone marrow stem cell transplant, the so-called BLT mice, developed by my former colleague at the UNC, Victor Garcia. And then the really quick and dirty model people can use for certain questions is the NSG Shield PBMC study. It's because the GVHD issue here, very quickly you are limited in the length of the experiment. So really, with all different model, you need to know the strength and the limitation of each model. So depending on what you need, we use all three different models for our study. So in this model, with the human stem cell, we can really study HIV infection and show viral replication, HIV pathogenesis with a persistent inflammation, tissue damage, and the CD4 T cell depletion, mimicking what's in the HIV infection. So in using this model, we really ask the first question is, we know HIV is an autoimmune-like disease. It converts host immune cell to cause damage. So the first question we ask is, which HIV target cell is helping the HIV to cause the disease? So we use the simple method of using antibody to depleting all different CD4 target cell and to see what happened to HIV pathogens. So essentially, that's a summary of 10 years work of 5 to 10 postdoc. You can see we discovered the pDC depletion really gives the most interesting phenotype. I'll explain later. It seems to lead to interferon production cause really persistent inflammation. Macrophage cause many tissue damage I won't talk about today. And Treg, ironically, during chronic infection, it's really helped to slow down the disease. If you deplete Treg during chronic infection, you make some disease in certain organ really worse. So pDC, let's talk about. So plasmacytoid dendritic cell was really a misnomer. If you don't activate them, they really should be called interferon producing cell. They are like a lymphocyte, produce interferon very rapidly, and they express high level CD4, higher than a macrophage or DC, just like CD4 T cell. They have two unique human-specific receptor, only expressing pDC, pDCA2 and T7. And the two interference sensing receptor Tau 7, Tau 9 in the endosome, one for RNA, one for DNA. And really what's unique about pDC is it expresses continuously RF7. So as a result, if you purely analyze the cell in vitro, each purified pDC can produce 1,000 times more type of interferon than any other cell in your body. And it produces very quickly. And it transitions to a dendritic cell, like here, the converting to dendritic cell, then lose the expression of interferon. So that's just if you think about the green line here, this really mimic what during acute infection, a green line look like for pDC suppression. Of course, interferon during primary early stage is important for every immune cell to function. So it's really important for initiating immune response. So I think it's been a search for decades by immunologists and virologists. I think in the late 90s, Yunjun Liu's lab from MD Anderson at the time and really discovered the human pDC, give the name and did much of the early work. First, Segal teamed up with Yunjun Liu and showed in HIV patient, this interferon producing cell really disappear from blood rapidly after infection. His conclusion is that they are depleted and impairing function. But later on, it turned out those DC, like many DC, if you activate them, they leave the blood, they migrate to the lymph tissue. That's where they cause a lot of damage. So to look at the pDC during chronic HIV infection, we infect the animal for 20 weeks. Then look at in the lymph node and spleen what happened to the pDC. And that's surprisingly, we find this pDC not only present at a good number in the lymph tissue, they constitutively express this red type 1 interferon continuously. And their antigen presenting cell function is very much impaired. So CD4D, class 2, and a lower expression. And the second part is if you label with BRDU for 3 days, you can see normally pDC are not labeled by BRDU. They die quickly, then replenish. But in HIV infection, the BRDU incorporation is increased in the tissue. And if you treat with the ART to suppress HIV replication, the BRDU is reduced, but the pDC persists with some self-renewing proliferation. So the question is this really a pathogenic cell? It's just a correlation study. So to prove it's a causal relationship, we have to develop a reagent because pDC was not well studied at the time. So based on these two receptors, I think initially identified by Yunjun Liu's lab, that this receptor are uniquely human pDC specific and not in the mouse genome. So it's easy to make monoclonal antibody. And using antibody and humanized mice, because very hard studying in vitro of a human pDC. The humanized mice turned out to be perfect for this study. So we develop antibodies, screen them for the ability to deplete human pDC in different tissue efficiently. We identify this 15B antibody, specifically and efficiently deplete the pDC in vivo. So the first question we ask is, what happens if you deplete pDC at different time before and after infection to see what happened to HIV? If you deplete pDC prior to infection, there will be no interferon induction, but after infection in 1 week. If you deplete at 6 days, they are fine. But you can see if you deplete pDC, HIV replicates slightly better at 1 week, but more or less plateau to the same level as other control group. If you deplete from day 6, you do not change as much. But if you look at anti-HIV T cell, that's the interesting part, is that during normal infection, you have some priming of anti-HIV T cell for interferon gamma and IL-2. Mark have no response because no priming. If you deplete pDC before HIV infection is like mark. There's no priming. If you deplete 6 days later, you get good priming. So this really what pDC is needed for the first 5, 6 days to prime T cell. And as we show later, they contribute to many disease progression later on. So use this antibody, but then we deplete pDC during chronic infection to see what happened to HIV pathogens. To make a long story short, essentially you have about 10 times more virus replication. So HIV's pDC is still antiviral, the interferon. But with 10 times more virus, you more or less have no pathology in the tissue. You can see the human leukocytes are rescued, both in number and in function. If you look at different tissue in the bone marrow, gut, spleen, it more or less back to normal, even with 10 times more virus. So that tells us the pDC is really playing a very critical role in HIV pathogens. And to make long story short, we test whether interferon, type 1 interferon, is contributed. We made antibody against IFNAR, and really to show this seem to phenocopy much of the pathology rescue, not all, some. So if that's unique for this humanized mice, you can see in this on the left, if you deplete pDC, the blue line viral replication to high level, but the pathology is gone. So there's no pathology and no inflammation. Actually, in SIV infected monkey, that's happened. In African monkey infected by SIV, you have similar viral replication as Asian monkey infection, but only Asian monkey infection develop AIDS, not the African monkey. And the correlation is IgG interferon. So the question is the pDC involved in this one? We are not sure. It will be a long, expensive experiment to prove that. But interestingly, really, human long-term non-progressor is very much like this one. The IgG signature will self-resolve even with virus replication. So just to move to something currently relevant in human patient, that's what happens is that with ART, you don't have detectable viral replication, and yet viral reservoir persists and inflammation is still happening halfway here. So we tried this in the humanized mice by using the same combination ART, no viral replication in the blood or in the tissue by flow. But if you look at their inflammation, it's still above background. If you see the reservoir still there, at a similar frequency as in some HIV infected patient, either by HIV DNA in the cell or by replication competent virus. If you stop ART, all infected animal rebound within 2 or 3 weeks, just very much like in human study. So using this model, we really asked the question, can we study how to control HIV reservoir by depleting pDC? So in this case, we infect animal, treat the animal with the pDC depletion or isotype control, and you can see the pDC is effectively efficient depleted, and the interferon signature on the ART is resolved. So a major inflammation-inducing factor is a pDC during ART. If you look at PD-1, you can see partially reduced by ART, but pDC depletion almost completely resolved with PD-1. Consistently, if you stimulate this T cell with the HIV peptide, although T cells are rescued by ART, they don't seem to function very well. If you deplete pDC, this T cells start to function by producing cytokine. And if you look at HIV reservoir, you can see HIV pDC depletion really lead to rescue or reduction of reservoir. It's not clear. It's just reduction. And so that's happening in HIV patients. So we take PBMC from HIV patient on the ART for 5-15 years. It's really good suppression of HIV. we treat this HIV-infected PBMC in vitro for 9 days, plus minus pDC depletion. So we have a toxin conjugate antibody can deplete pDC. Then after 9 days, we stimulate them with HIV peptide and to see what their function. You can see either pDC depletion or if not blockade, we will rescue the T cell number and T cell function under this condition. So the same pathway is functional in HIV infected patient under. So we also, as I said, test, can we achieve HIV cure? In this case, we block interferon with the IFNA antibody. And then after treatment, we remove ART and to see how long does it take for HIV to rebound. You can see the black isotype control is like now 100% rebounding in 3 weeks. For the if not treated patient, it seems to have a delay, but it's really not cure. So you can see HIV reservoir is really reduced efficiently, but that's enough to rebound. That's consistent in HIV patient. Even you have 3 log of reservoir reduction, they still rebound. So it's really challenging. So achieving using the this targeting this plan, I thought 3R, we achieve resolved inflammation and rescue anti-HIV T cell and reduce HIV reservoir. So back to the beginning. So how does HIV induce end organ disease? The current idea, I think, is really the residual inflammation probably induced by a HIV reservoir, because HIV reservoir in the tissue have random firing and they're producing virus. This virus will activate pDC very efficiently. Even without production, pDC uptake viral particle get activated. The second part really other cofactor, virus infection, alcohol, fat diet, and the toxicity of ART. So that's all contributed to this disease. So we are currently focusing the lab really to study how to treat HIV associated end organ disease. I'm not quite brave enough to try to claim we can cure HIV. It will be very challenging. So here's the example. If you model in the humanized mice HIV infection, if you suppress with ART, there's no viral becoming detectable. Yet you have ALT level, you have liver injury associated with liver fibrosis. So ART plus HIV lead to liver injury. And this injury, if you either deplete pDC or block IFNA, you almost completely significantly block the liver fibrosis. So this pathway in this model is important for the liver disease. This is the liver disease. You can see you need both HIV and ART to induce liver injury here. So based on this find that really we are currently pushing the one of the antibody we developed to confirm this finding in SIV or in people live with HIV, hopefully have a proof of concept study. So during ART treatment, if we deplete pDC, can we achieve first resolution of inflammation to reduce end organ disease? Second, do we control the reservoir and delay the rebound? That's, unless it's cure, really, the rebound delay may not be as significant, but will be interesting to study. So that's where we are for the HIV study. So just to go to the next phase of a pathogen study, for HIV chronic stage, especially with ART, it's really a very, very low level precision cytokine production contributor to disease. You can think about a cytokine storm versus cytokine drizzle. So for COVID, everybody knows the cytokine storm now. And in this chronic condition, really it's more like a drizzle, but for very long. So this is from Wikipedia was the definition of the plum rain. It's really a stationary precipitation persistent for months, leading to rotting of everything inside. So that's just to give you some early poem by Dufu about this straw thatch is loose and easily soaked. Clouds, fog are dense and hard to break. So really, this persistent fog and cloud is what causing all the damage. So what do? So we stop the rain or clean up what inside is what we think how to treat this kind of disease. So before I move to the next phase for disease treatment, I'll give you a dose of mechanism study. So how we study the mechanism. So the first question is, how does HIV infection induce this disease-associated pDC in the tissue? So this years of our work and another group from New York really identify multiple HIV-encoded factor work together to program this pDC. One is the HIV envelope through CD4. So CD4 interaction is critical. They go to the early endosome, the top 7 is still active. They can recycle and get continuous activation. The second one is really the VPU. It targets tethering for virus to be released. But tethering is also ISC to shut down the pDC through the ILT7. That's the feedback pathway. HIV degrade tethering through VPU. Another one, we and the Xiong lab, we work together, identify VPR HIV. Actually, target TET2 is a DNA demethylate. It's recently He also showed it's RNA demethylate too. So it's very important one in regulating gene expression. So one thing we showed that it actually lead to constitutive expression IR7 in pDC. So that's why pDC is so efficient in responding to stimulation by virus. VPR together with HIV viral particle target this pathway, really delay the response of pDC to virus infection. So that's a lot of virology and cell biology. I won't bother give you too much detail. Just to show that we did this by viral genetics. So we use different viruses with different genetic potential. By doing a recombination between different viruses, we map the pathogenic factors to be here. One is major binding CD4, activates pDC with no fusion, just go through endosome that caused immunopathology. And CCR5 binding itself can trigger cell death. And the fusion most likely is a major factor comes out. So with this mapping, we really think the pDC activation is an interesting immunological target for us to follow up to the study. So here's an example, a diagram to show you how that's CD4 mediated endocytosis different from sialic acid endocytosis. When flu infect pDC or deactivate pDC very efficiently, go all the way to the late endosome and stop interferon production, induce NF-coupled beta. For CD4, it targets to the early endosome, get pDC activation. But the tau-7 recycle, and as a result, HIV-infected pDC can be re-stimulated continuously as a result. And they're all determined. Okay, so what's the mechanism? So how does HIV really induce the DAPDC to cause immune suppression and tissue damage? So the question here, because we had this model, we can really study different conditions and compare to the multi-omics functional assay with the different condition. And that's again, 5 to 10 years work summary. We showed that there are at least 5 different pathways is upregulated by the disease-associated pDC to suppress T cell. First 3 I call, first 2 are pretty boring. So predicted PD-1, PD-L1, Treg, TAM, myeloid cell, and some impaired DC. The lymphoid tissue fibrosis is interesting. I won't have time to talk about it. It seems to be another mechanism. Today, I'm going to give you some of the data to show that number 4 and number 5. somehow this pDC interferon pathway to really reprogram the mitochondrial activity in T cell. So they use glycolysis over oxyphos, and as a result, it's really not functional well. The number 5 is wrong. This is number 5 is really the key CD8 T cell for controlling a chronic antigen response, that is the stem-like CD8 T cell. So to study first, what's the cell important for controlling HIV reservoir? Liang and Guangmin Li, they did the experiment separately. They deplete pDC or block with Ifnar at the same time, also deplete human CD8 T cell to see what happened to HIV reservoir control. You can see in both cases, pDC depletion or Ifnar blockade control reservoir dependent on CD8 T cell. Did CD8 or not? So CD8 T cell are important in this case. Then Liang Cheng isolate CD8 T cell from this isotype control Ifnar blockade one and did a RNA-seq analysis. And we showed 164 different genes suppressed by this Ifnar blockade. They are all interferon stimulator genes, immune activation genes, cell death gene, and T cell exhaustion genes. So that's interesting. But another interesting class, 140 genes, actually upregulated by interferon blockade. And those seem to be all involved in mitochondrial activity, the oxyphos pathway. And to test this defect, I think we showed if you take the T cell from HIV infected humanized mice from tissue, their mitochondrial potential, membrane potential is impaired by HIV infection. If you use ART, suppress HIV, you rescue T cell, they are still defective in JC1, this is a mitochondrial membrane potential. If you block interferon, they are restored. So using a low throughput assay, Liang, essentially we take the spleen from this HIV infected animal on the ART, then treat them in vitro, stimulate with CD3, CD28. You can see mock infection works well. The HIV infected animal, the T cell do not respond to TCR stimulation. Then under the condition, Liang just treat this culture with a different inhibitor, different glycolysis inhibitor to see which one can rescue CD4, the T cell response under this condition. Interestingly, 2DG, the 2-deoxyglucose, is the one most efficiently restore T cell function. Metformin, which target a similar pathway, seem to work at a lower, at a higher dose. It works better on CD4 T cell, which is a different story. So anyway, fortunately, the 2DG has been used in cancer treatment in vivo, and we use a dose which is not toxic to animal in HIV infection, and to see that if you treat with 2DG under this condition, with ART, you rescue central memory T cell, which is defective in HIV infected patient on the ART. This one seemed to recover them. And PD-1 is further reduced, and the HIV response to HIV peptide is restored. You can see they respond to T cell again. And if you look at the HIV reservoir, they are efficiently suppressed by HIV if you activate them. So in this case, really HIV through the ART together or the interferon really impaired the mitochondrial activity in this pathway. To preferentially use glycolysis in T cell and with less than optimal function. If you treat it with 2DG, the restored is somehow convert into a more oxyphos pathway. And how this thing work is we are currently studying. There's a lot unknown about how metformin and 2-deoxyglucose work. And fortunately, I met Ed Pierce from Johns Hopkins, who's really expert in immune metabolism. he published a paper a couple years ago showing if you starve the T cell transiently, normal T cell with glucose restriction, the mitochondria is restored better and to function better. We think that might be the pathway the 2DG is doing to stress mitochondria to restore the function. So that's the highlight of the story I just forward to you. I think a couple of years ago, Haitao Guo and my postdoc Qi Wang, together with Jenny Tin's lab, we published a paper showing 2DG metformin actually reduce HY replication in CD4 T cell data. So really it makes interesting to test how this pathway targeting HIV patient will work on patient. So for the next part, really the stem cell like stem like CD8 T cell, I think roughly Matt and others have recently shown this during chronic infection or in cancer patient, the presence of this PD-1 positive, TCF1 positive T cell, this TIM3 negative, a stem-like T cell that respond to anti-PD-1 immune checkpoint. The really terminal differentiate TIM3 positive TCF1 negative cell fail to respond, really. So those cells are critical in the tissue to fight a persistent antigen in virus or tumor antigen. So the question we've asked is this tumor antigen, is this pathway important in regulating the stem-like T cell? I was at the Emory, talked to Rafi for almost a whole afternoon to design the experiment right before the pandemic. Unfortunately, we were delayed. So this here's a catch up what our discoveries. So using this model, we can study what happened to central memory T cell and to stem-like T cell. So to give you a quick summary, really deplete pDC rescue central memory T cell, and in both number and percentage, and also the stem-like CD8 T cell is rescued. If you look at stem-like T cell correlation with the HIV reservoir in this treat animal, you see a very significant correlation. R is 0.8, P is really 0.001. You have more stem cell, you have less HIV reservoir. So how about this pathway in HIV infected patient? So we got HIV, PBMC from HIV infected patient and stimulate them with HIV peptide with CD28 ex vivo, which is fresh. And Guangmin Li in the lab developed this 23 or 27 color flow cytometer assay and identify this T cell with a polyfunctional activity expressing this multi-cytokine, and to show that this polyfunctional HIV reactive T cell are mostly the stem-like T cells, so they are PD-1 positive, TIM3 negative cell. Then if you analyze those cells differently to see healthy donor was HIV infected PBMC, stimulated in this case with CD3 antibody, to see what happened to the multifunctional T cell. You can see, compared with healthy donor, the HIV stem-like T cell actually impaired in function. So the question is, can we rescue this with a pDC depletion? So we use the human PBMC in vitro, do a 9-day co-culture, and look at the function after stimulation. You can see the polyfunctional T cell are elevated by pDC depletion. If you look at even the positive cell for their cytokine mean fluorescence intensity, the pDC rescue their not only number, also their intensity of cytokine induction. The same for stem-like CD8 T cell. So in this case, we showed HIV, the pDC seemed to impaired stem-like T cell to contribute to HIV persistence. So if you deplete pDC, you expand this stem-like T cell in number and function, probably control HIV better. But the obvious question here is, are this cell now become more responsive to PD-1 immune checkpoint inhibitor? So we test this idea is using the HLA2 positive HIV infected patient PBMC. So A2 positive, we can do tetramer. So we do the same assay with the tetramer peptide, and then show if you deplete pDC, you really rescue this tetramer positive cell in percentage number. This is percentages, this number. And each individual rescue partly, but combination of pDC depletion and PD1 blockade, you rescue cooperatively. If you look at their tetramer positive cell for polyfunctional T cell, you can see the combination rescue their polyfunctionality. And the same for total stem-like T cell. So in this case, we show HIV specific polyfunctional CD T cell are impaired, and depleting pDC rescue that and make them more responsive to pDC immune checkpoint inhibitor. So that's the HIV part. So as I said, we use an HIV to really dissect human inflammatory pathway. We think up to now, we conclude we identify this disease-associated pDC mediated inflammatory pathway. And we just for simplicity, we call it inflammation pDC. In this case, the trigger in this case is HIV. The effector cell are the pDC associated disease pathogenic pDC. One of the major mediator interferon, type 1 interferon, but it's not all. We know some of the phenotype are not phenocopied by interferon blockade. And mechanistically, we identify 5, 6 different ways this can really lead to immune suppression. Therapeutically, we have developed antibody for pDC, for IFNAR, and for metabolic reprogramming. So then the question is this pathway used by other human inflammatory disease that we can study? So if you look at acute inflammation, where they are engaged pDC, there are many things. So pDC is very sensitive to many different type acute immune challenge. So the real problem is, at most of the time, they are doing good things for trauma injury, burns, wound healing, and they are really good. pDCs help to suppress that, very much like they help to prime anti-HIV T cells. The real question is during chronic inflammation, this disease-associated pDC may contribute to all these things, which at least for HIV and maybe even without HIV. So we show the chronic infection is beneficial if you block this pathway. And I personally had time to show, we show in the MASH, that's the high-fat fed animal model, where block this pathway also reverse a liver disease. And of course, during the study, Biogen have developed a similar antibody. They already now in phase 3 clinical trial to show SLE or lupus patient benefit, really one of the better treatment for 50 years to treat some lupus indication by block deplete pDC. But today I'm going to show this pathway is also in some type tumor as in the tumor microenvironment. And what we have done is HCC, the liver cancer, and the triple negative breast cancer. In ovarian cancer, although pDC there, it did not work. So it's not like everything we look at work. The multiple type thing, depleting pDC did not work. I will not show you the next result, but just the next, I have probably quickly 5 minutes or so to show in this pathway, is the tumor engaged the same pathway and with similar TME to suppress T cell by this 5 different pathway. So can we target the tumor-associated pDC to program TAE to again achieve similar thing as this? To make a long story short, probably I won't show it if it doesn't work. Right? So you can take that as a take-home message. But here in a liver cancer patient, there's a multiple studies showing if you have a intratumor pDC, you have a very bad prognosis in liver cancer patient. So we build the model to see, can we study this tumor pDC in human tumor in the humanized mice. So I built this one I call the HIT mice. Essentially, we build with humanized mice with the human stem cell reconstitution first. Then we put in a human liver cancer cell line or PDX in there. Then we can study a human tumor microenvironment with human immune cells. So one interesting finding here is if you have humanized mice in ASC mice, the tumor grew better. So the human immune system promote the tumor growth in this model. And if you look at the tumor environment, you have a lot of pDC in the tumor. It's like 5 times more than the lymph in the spleen. And this tumor-associated pDC are partially activated, very much like in HIV infected spleen. They are partially activated. And the tumor-bearing pDC in the tumor-bearing spleen is not activated. So it's really a tumor-specific. And Treg is associated with pDC, and the stem-like T cell is very low in those tumor microenvironment, and we exhaust T cell are high. If we deplete pDC in this tumor model, you can see pDC is very efficient depleting in the tumor microenvironment. MDC is not touched. Type1 interference is suppressed. Treg is suppressed because Treg really in the TME is induced by pDC. Stem-like T cell is up in number and in function. And if you look at the mitochondrial activity of the T cell, depleting pDC also rescue the intratumor CD8 T cell mitochondrial membrane potential. So then the question is, can we show combination therapy with stem-like T cell to the combination? We did this in multiple human cell line and mouse cell line to show that indeed, if you deplete pDC and PD-1, you have a cooperative suppression tumor and survival. In this case, the tumor survive better. And this, of course, early, and we are repeating the more and more. So that's the summary of what's in the tumor. You can see pDC, in addition to suppress T cell, somehow promotes tumor growth by an unknown mechanism we currently don't know. And the effect on stem-like T cell can be used to combine with PD-1 treatment, have a cooperative treatment for immune checkpoint. So we are also think about how to use this combination with CAR T cell, and we'll have time to show you today. Just a quick summary of what our study with triple negative breast cancer. If you look at the tumor infiltrated pDC in breast cancer, you can see similarly, if you have breast cancer, you have pDC, the prognosis is very bad in this patient. And we have done similar work just to give you a quick clinical trial-like study in humanized mice, where in this case it is a triple negative breast cancer HIT mice, where we...
Lishan 能看到我吗？听得到我吗？
Yeah. Is Li Qiang, are you talking to me?
No,
Okay.
No, no.
Yeah. So in this case, we follow what's ongoing at the UNC. We have a clinical trial to treat the triple negative breast cancer with a Abraxane for 1 week, followed with a PD-1 antibody for 3 weeks in this patient. So I use the same protocol in humanized mice to do a Abraxane for a week, then use the pDC depletion for two weeks. You can see each single treatment suppressed by 50%. Combination synergistically suppress tumor growth. What's really more interesting is on the right. Abraxane, the chemotherapy, although reduce tumor, they enhance metastasis to the lung and to the liver. The pDC alone did not enhance metastasis, but combination really prevent the metastasis of Abraxane. So that's really something we are pushing, how to push to the clinic when the antibody is ready to be tested. So this is the summary of multiple anamorpho. BRCA enhanced metastasis, pDC did not, and pDC suppressed. So this is the summary of I'll stop here and just tell you that this disease-associated pDC really involving multiple inflammatory tissue and disease, including infection, autoimmune disease, and in tissue fibrosis, especially in the liver and in cancer. This is my lab. The red are my University of Maryland lab, and the blue are the former UNC member who left the lab, contributed to this disease. And in particular, Li Guozhang, now at the Institute of Biophysics in Beijing, really started the whole pDC project in the lab. And Liang Cheng at the Wuhan University, and Kovalyev, Qi Jiang, Natalia Qi Jiang, Zheng Zhang, they all play very important roles, the early work. And James, Guangmin Li, and De Gui Geng are the three postdocs currently, and the research faculty are doing the current work. Jenny Tin from UNC Chapel Hill really support us in many of the early study work, and Yue Xin doing the molecular mechanism work. Yunjun Liu, as the pioneer of the pDC study, we really work with him closely at the beginning since his MD Anderson days. And Alex Plas, Brad Zheng, and of course the Emory group, including Raphael Matt, have been providing great help. Brad Zheng and Nilu providing human study, and at the UNC at the Maryland, Yang Liu, now in Encore C4, have been involved in some of the early work, and we are working with him for some of the immune therapy for cancer tumor study. And Sham Cotton-Leo is the clinical person, provide some sample, and we are working with Li Qinhui to study some of the liver metabolism, hopefully soon. And CMV Wang from NCI, and Wang Jinhe and Ed Pierce from Johns Hopkins are all involved in some of the study. Just a quick conflict disclosure. So this is a company, beside the NIH support, the company funded, and the company I provide consultant for. I'll stop here and be happy to answer any question if we still have time.